So welcome. We are going to get started now. I am Marcella Moss. I'm an assistant professor at Harvard Medical School and director of the Cellular Immunotherapy Program at Massachusetts General Hospital. But I'm better known as Carl June's former graduate student. Thank you all for joining. These lectures are brought to you by the Dana Farber Harvard Cancer Center and the Leukemia and Lymphoma Society. Now, before I get started, I'm going to just give a brief introduction to Dr. Carl June, as I'm sure all of you know him. I've had the privilege of knowing Carl June for over 20 years now, and his story and career never cease to amaze me. Carl attended the U.S. Naval Academy in Annapolis as an undergraduate and Baylor College of Medicine for medical school. In his last year of medical school, he worked in a laboratory at the World Health Organization, and then returned to training in internal medicine at the National Naval Medical Center. He completed his fellowship in oncology at the Hutch, where he worked with E. Don Thomas and trained in bone marrow transplant. It was then that he discovered the function of the CD28 co-stimulatory molecule in human T cells, and he made an agonist antibody to CD28 that would go on to be a bedrock of his career. After his fellowship as a newly minted oncologist in the early 1980s, the Navy decided that he needed to work on HIV instead of cancer. This background, though, in bone marrow transplant, coupled with his work in co-stimulation and ultimately what he learned in HIV, now seems like it was fate that he would be the one to design and translate the breakthrough therapy that we now know as CAR T cells. Dr. June has touched every aspect of what makes gene-modified T cell therapy possible in human patients. While he was in the Navy, he coded magnetic beads with his antibody to CD28, along with an antibody to the T cell receptor. And he discovered that these artificial antigen-presenting cells served to not only expand T cells past the Hayflick limit, so they had stem cell properties, but he also made them transiently resistant to HIV infection. This work led to some of the first trials of adoptive immunotherapy, where he tried to restore T cell counts in AIDS patients. In 1999, he moved his laboratory to the University of Pennsylvania, and he set up a GMP facility to prepare these bead-activated, co-stimulated donor lymphocyte infusions for patients with HIV, and then extended this platform for patients with cancer. Over the next decade, Carl developed zinc finger nucleases to gene edit the CCR5 co-receptor for HIV, and chimeric antigen receptors targeting HIV, the solid tumors, targeting mesothelin, and the B-cell antigen CD19. The first 2 patients treated with CD19 CARs went into complete remission, and a third had a very good partial response. If you look at the impact of his 2011 New England Journal of Medicine publication that described these findings, you can see that this is the inflection point for all of cellular therapy. I can't overstate the impact of this work on patients, on the field, and on scientific progress. More recently, Carl has turned his attention to T-cell genome engineering and has recently published the first clinical trial using CRISPR-Cas9 edited T cells, which I think we'll hear about a little bit today. Carl has received numerous awards for his research, including the Lifetime Achievement Award from the Leukemia and Lymphoma Society, the sponsor of this lecture series, the William B. Coley Award in Tumor Immunology, the Philadelphia Award, the Lloyd J. Old Award, and the Ehrlich Award, which he shared with Jim Allison, and the Karnofsky Award, and is a fellow of the Institute of Medicine and the American Academy of Arts and Sciences. In 2018, he was one of Time Magazine's most influential people, and what's especially notable is that he was nominated by the first pediatric patient who received his CAR T cell therapy. Carl June is inspiring to all of those who meet and interact with him, and I'm glad that even in this era of social distancing, all the participants can meet him virtually. He's going to talk to us today about T-cell genome engineering in the clinic. And I just wanted to bring up 2 quick technical things. One is that if you have questions during his talk, please type them into the Q&A button at the bottom of your screen. And also for your information, the slides will be posted on the website in the next couple of hours to days. Thank you very much. And with that, I will turn it over to you, Carl. Thank you for joining.
So thank you, Marcella, for introductions, and and for David Weinstock and Nellie for organizing this. My voice is a little raspy as I'm just in the 2nd day of convalescing from my own case of COVID, and I'll try to weave some of that into my discussions here about cytokine release syndrome later today. So my conflicts are listed here with research, sponsor research, and IP with Novartis. And Founders Equity and Community Therapeutics. I won't be discussing their trials today. Cancer has a deep relationship to the immune system. If you look in this, an article last year, a review of different mechanisms of immune tolerance, there were 10 listed. When I was in medical school, there were 2. One was thought to be the thymus, and then we didn't know about the rest of these post-thymic mechanisms. But now there are a number of mechanisms that occur at the thymic level and then in the periphery. And what you'll find if you study in detail tumor microenvironments is that various tumors phenocopy basically all of these basic mechanisms. Right now, a major part of the work in the field is understanding the mechanisms that tumors induce T-cell exhaustion, and also the issues of regulatory T-cells, and circumventing that is a major issue for the field. But all of these have been co-opted at various times by tumors and in various histotypes of tumors. A few years ago, Michelle Sadelain and I wrote about sort of the status clinically of T-cell therapies, and the 2 main kinds are showing here. One is T-cell receptors that are HLA linked and target the intracellular pool of nucleic acids in tumors, and that become peptides. And then the other is chimeric antigen receptors, or CARs. One major difference is that the CAR targets are surface molecules. They can be glycoproteins or even glycans, but they are not MHC presented, and then they don't care about what your HLA type is. And whereas these are restricted to your HLA type and C intracellular targets. So non-overlapping set of targets and very different mechanism of actions that I think will, in the end, will be probably synergistic because they can each have different resistance mechanisms. Let's see. So there we go. If you look at the genealogy of CAR T cells, it goes back basically to the era of recombinant technology with Asilomar, and then the ability to engineer cells genetically initially with retroviruses that Michelle Sadelain did in Dusty Miller's lab a long time ago. Adoptive T-cell therapy began in the field of bone marrow transplants with allogeneic cells, and then with TIL cells at the NCI for melanoma, and leading to CAR T cells now. And then the CAR idea to make an MHC unrestricted cell, was first published in '87 with data in Japan, and then later at the Weizmann Institute. And there have been a number of design iterations that have finally led to what we now call 2nd generation CAR T cells that are FDA approved. And not only can you, retrain T cells to target virtually any surface target on healthy cells or tumor cells, but you can also take advantage of the intracellular signaling domains in CARs. And that leads to the changes in the fate of those T cells. So if you start with a naive T cell, a CD8 cell that expresses a CAR molecule, in this case for CD19, and then activate that CAR by culturing the T cells with leukemic cells that express CD19, the CAR T cells will kill the tumor cells, but the end up culture at the end with the case of a CD28 signaling domain will be cells that are effector memory cells that have changed their glycolysis and metabolic energy sources to non-mitochondrial glycolysis, whereas the T cells that had the 4-1BB signaling domain will end up enriched in central memory cells that use fatty acid oxidation and have active mitochondrial biogenesis. In fact, the genes encoding mitochondrial biogenesis are turned on selectively. So you can reprogram both the specificity of the cells and metabolic fates and end up having cells that have long-term or short-term persistence in patients, something you can't do with, say, for instance, a checkpoint therapy where you unleash cells but don't specify their fate. So we first began treating patients in 2010 with the 4-1BB based CAR, and David Porter reported the first patients in 2011 who had had chronic refractory CLL. Two of the three patients entered remission, which is ongoing until today. And we've treated now in the field, treated virtually all B-cell malignancies and found activity. In our case, they've all used autologous cells cultured with a 10 to 12 day culture process, a lentiviral vector to genetically modify the cells, and both adult and pediatric patients. Our first pediatric patients were treated in 2012, and that's where we really learned a lot about cytokine release syndrome. And that led to FDA approval in refractory ALL in age 3 to 25 in 2017. The first CLL patients, as I mentioned, treated in 2010, we had a 5-year follow-up of them published in 2018. And both patients here, shown 1 and 2, have had a PK of the CAR cells, which shows CAR memory cells out to 5 years. They now actually are now 10 years out in our unpublished data. So they maintain functioning CAR T cells after a single infusion, but they have an acquired immunodeficiency. Both patients here have no B cells that have rearranged IgH, so they're all in the germline configuration of their B cells, which meant right now, if you had a vaccine that worked for COVID-19, you wouldn't be able to vaccinate these patients. Now, interestingly, human plasma cells that are memory cells do not express CD19, and they persist in these patients, adult patients who've had a full memory repertoire of antibodies induced through childhood infections don't have an increased incidence of opportunistic infections, even though they have this ongoing B-cell aplasia. But this is clearly one of the areas where there will be innovation to have inducible CAR molecules that could let B cells regenerate after an induction is given. So this shows a principle that you can have a living drug, a single infusion can stay active for now a decade and induce long-term remissions in patients. In those first 2 patients I showed you here, they had peaks in their PK of the CARs at 10 days and 23 days after infusions. And we noticed in the first phase 1 trial that, where there's a 57% overall response rate of CRs and PRs, that there's a broad range of time to Cmax here from 10 days out to 50 days in this patient, patient 10. We also noticed a biomarker of clinical efficacy, which is in the CAR molecules as plotted semi-logarithmically here of copies of the CAR per microgram of genomic DNA. The proliferation in these patients out to a year here correlated with the responses, and you can see non-responding patients did have engraftment, but log orders of magnitude lower peak, Cmax levels. And we've seen this not just in CLL, but in ALL and in myeloma, and now in some solid tumors, so that we can say that the in vivo engraftment and proliferation of CAR cells has been a predictive biomarker of clinical benefit. And patient 10 that had that delayed peak had a really interesting finding when we did a bedside back-to-bench experiment. It turned out his CAR T cell was integrated into the gene TET2, which works epigenetically to control cytosine methylcytosine on DNA, and TET2 can deoxygenate that and hydrolyze it to make 5-hydroxymethylcytosine and further oxidize that. So it regulates epigenetic marks, and it's been known that this can change stemness in cells, and that it's also, as shown by some studies out by Ben Ebert at Harvard, it can lead to clonal hematopoiesis if it occurs in your stem cells. This actually can be a good marker, as opposed to some other genes like DNMT3A, which if it's mutated has a higher incidence of leading to AML. So we found in that patient 10 that the CAR T cells that grew out in him were the progeny of a single cell that had one integration of the CAR in chromosome 4q24, which is the locus encoding TET2. And that CAR was intronically between exons 9 and 10. And that then prevented the dioxygenase from being expressed in that patient. So it was a loss of function and due to premature stop codons that we described here. So we learned a lot. Lentiviral vectors integrate mostly into active transcription units. When we activate T cells in vitro, there are many genes that get integrations, which oftentimes can lead to loss of function. And we found in this patient 10 that the progeny from a single cell led to the eradication of the cells, the tumor in him, and now ongoing complete remission. And that says potentially that the lowest effective dose of a CAR cell could just be a single CAR T cell if it had massive proliferative potential. So we found in that case, in patient 10, that the unintentional knockout of TET2 was responsible for enhanced CAR T cell function and restrained differentiation so that they maintained more stemness. And then we've since found that we can do this, and the details are in this paper. We can intentionally do this with using CRISPR-Cas9 technology and phenocopy this in healthy donor T cells. Lost in the madness of what our COVID epidemic has done, a paper came out in February from Rick Bushman's lab and our lab, where we now look not just at patient 10, but at 40 patients in detail and integrated all the annotations and annotated where the CAR T cells had integrated in their genome. So we looked at the long-term surviving CAR T cells, and we used this sort of pipeline to do this. We took patients at baseline and looked at the integration samples of what was infused into the patient and then the long-term surviving cells in the patients, and found a number of genes were either lost in abundance or enriched in the patient's cells compared to that infusion product. We found 6 examples of TET2, so that's not just an isolated incidence, but it's rather frequent. Although we didn't see clonal outgrowth as we had in the case of the patient we showed, but they are dominant integration sites, and there are others that you could refer to in this patient as probably enhancing fitness of cells in patients or decreasing fitness. So it's a really very unique opportunity to be able to study with this reverse genetics approach, because the cells are genetically modified. And we're basically doing genome-wide, if you will, CRISPR mutagenesis of the human genome, at least in T cells in these studies. We found, that there was cytokine release syndrome at the peak, and this has been seen both with the Gilead CD28-based CAR as well as the 4-1BB-based CAR that Novartis has marketed. And tocilizumab, marketed by Roche, is co-labeled for this, for both CAR cells, and it's given at the time of cytokine release syndrome to attempt to abort the need for advanced medical care. And this has come into the fore now because there are international trials asking if the same reagents, blocking hypercytokine reactions in patients with COVID-19 might also benefit from this approach. And we have trials on at our institution, and I'm sure you do in Boston as well. There are other approaches for cytokine release syndrome primarily for CNS penetration, corticosteroids, although they are not as effective, but for patients with primary CNS dependent adverse events, they may be first choice. You can also block cytokine sinks, although there's differences between directly targeting IL-6 versus targeting the IL-6 receptor, which is what tocilizumab does, and the Regeneron-based approach. Because IL-6 signals three primary mechanisms: one, the classic receptor, and then other trans-presentations mechanisms, and there are differences on how an IL-6 directed reagent targets compared to the IL-6 receptor directed antagonist. IL-1 antagonists are being tested, as well as mechanisms to either kill CAR T cells or to turn them off, or also basically to remove cytokines from patients using apheresis procedures. So with that, I'll turn now away from bone marrow tumors, where there's very active trials for nearly every bone marrow derived malignancy you can think of, both using autologous cells as well as allogeneic and third-party derived T cells and NK cells, to solid tumors, where the major challenge is. So 90% of all the cancers in the US, are solid cancers, and about 10% are bone marrow derived tumors. And there are some targets that have been validated through other mechanisms that are now being tested for CAR targets listed here, and then others in various states of credentialing where there's basically a difference between healthy tissues and the malignant tumor as to whether they can be drugged. And the therapeutic index in these is really only determined at this point now via clinical trials. The animal models have not been very good at saying whether one could target these targets on solid tumors, because they're not, with exception of a few targets, they're not the dream target, where in the case of CD19, we had patients with congenital immunodeficiencies of CD19 and mice as well, and they could be healthy. And it was a well-tolerated immunodeficiency. In the case of many of these targets, we don't have that information. So if you go through clinicaltrials.gov, you'll find more than 400 trials with CAR T cells now around the world, and that most of them, many of the great abundance are in China, with the US second in abundance. And at least as of in February, the #1 targeted site was brain cancer, with a variety of targets being tested. But you can see basically in whatever your favorite tumor is, there are CAR T cell trials that you can find in various states of being done, both in academia and with a very active biotech, as shown on this side. So at the University of Pennsylvania, this is an example of some of the CAR T cell and TCR trials that we have. I'm going to come back to a trial targeting York ESO1 shortly. We have CAR targets targeting in glioblastoma, initially was led by Marcella Moss when she was back at Penn, and now by Donna Rourke at the University of Pennsylvania, targeting the splice variant EGFRB3 with a PD-1 antagonist. We have patients being treated with a glycan targeted CAR. The epitope is TNMUC1 in a basket trial that's opened in December. We have targets led by a trial targeting the serine protease fibroblast activation protein that Steve Albeld is conducting at Penn. And this is a trial to target the tumor stroma. Because the fat molecules on carcinoma-associated fibroblasts, which is oftentimes overexpressed in solid tumor microenvironments. We have 2 trials open targeting mesothelin, and there are probably 5 trials around the world doing this now. It's a GPI-linked protein. It's overexpressed on all serous ovarian epithelial tumors, as well as pancreatic and mesotheliomas. And then we have a trial targeting prostate cancer, castrate-resistant with prostate-specific membrane antigen, and a dominant negative receptor to TGF-beta. So many different trials underway, all early stage at this point. What I'll turn to now is our trial using genome editing. You know, there's a number of strategies to potentially increase the efficacy of T cells. I mentioned how it can happen basically through random integration with lentiviral vectors and knockout of TET2. There's a report now, and that happened with Sybil as well. Here, we first did this intentionally using zinc finger nucleases in patients with HIV, and we knocked out the gene CCR5 back in, and reported that in 2014, and found that we got when we did that, we had long-term engraftment of cells. It had a single gene knocked out from NHEJ DNA repair processes. And we then began in 2016 to 17 to test CRISPR-Cas9 technology, which is clearly more flexible and allows multiplex editing, whereas that's very difficult with this platform. And so my colleague and I, Yangbing Zhou and I, looked at this initially, as many other labs did, in T cells using various approaches to express multiple guide RNAs in T cells. And in this case, we made T cells that were had knocked out HLA class 1 through beta 2 microglobulin and the CD3 T cell receptor chain, so that the cells became CD3 negative and HLA class 1 negative. And we also, at the same time, included guide RNAs to PD1 and CTLA4. So this is 4 different knockouts with Cas9 electroporation, and we got about 33 to 35% knockout of PD1 and CTLA4, so double knockouts in post-thymic T cells. When we went 5 and above, we found decreasing efficiencies, but we could get very efficient knockout, not shown here, up to about 90% with optimized conditions. And shown here is that in addition, we could make CAR T cells work better by knocking out FAS. And so when CAR T cells target a leukemia cell that expresses CD19, this is a wild type CAR T cell that does this. And if it has FAS knocked out, we get better proliferation due to decreased apoptosis. So there are both strategies to enhance the proliferation of CAR cells, as well as decrease apoptosis upon target engagement that could potentially be done, you know, with Cas9 engineering. And so for our preclinical trial, you know, and to go into the initial testing of this, we decided to test and knock out the T cell receptor alpha and beta genes in order to decrease mispairing with a transgene that would express chains that would target the cancer testis antigen NEORK-ESO1. And that we also want in the long run to see if that will decrease off-target effects from autoimmunity and cross-priming. In addition, we knocked out PD1, as many other labs have now done, because we found out that increased the efficacy of these cells if they had the knocked out endogenous TCR and then also PD1 being knocked out. So we have 3 different guide RNAs that have been expressed in T cells, along with Cas9 to make multiplex editing based on this technology in 2016. And that was published in February in science than on our first 3 patients. And what I'll say is this is technology that we had in 2016. I'm sure many of you in your labs now, including our own lab, can do this at much higher efficiency than we're able to do with the in vitro transcribed RNA and the Cas9 lower fidelity reagents that were available back in 2016. So this is an inevitable example of the technology racing ahead in the laboratories much faster than it can be translocated and translated into patients. So the trial design is a basket trial to target NEORK-ISO1, which is a cancer testis antigen expressed on a number of different kinds of cancer, and then triple edited at the T cell receptor alpha, beta, and PD-1 loci. And we tested a single infusion, so at flat dosing of these autologous T cells. The basket trial design was for patients who had either had NEORK-ISO1, and it turned out we had, in the first 3 patients, 2 myeloma patients and 1 patient with a sarcoma expressing NEORK-ISO1 and HLA-A2. So single dose, we're not allowed to do dose escalation or multiple dosing. Basically, the trial was designed to ask the safety and feasibility of this, not efficacy questions. And the manufacturing process is shown here. It involved apheresis, autologous T cells that were then electroporated with Cas9 protein, nucleic protein complexes with the guide RNAs, and then cultured for 11 days and harvested, and then subjected to a number of release assays. The lentiviral vector that was introduced following the Cas9 knockout is pretty standard in our experience. It was the alpha and beta chains separated by a 2A self-cleaving sequence, and lentiviral vector with a EF1 alpha eukaryotic promoter. So the rationale of this trial was to ask if multiplex editing could be safe and feasible, and removing the endogenous TCR could increase efficiency of the transgenic TCR and hopefully reduce off-target effects and autoimmunity. And then this came up after our trial was well underway in regulatory approval, which was a report from Jürgen Ruhlen's lab in Germany saying that in mouse T cells, PD-1 functions as a haploinsufficient tumor suppressor. We've not been able to prove this in human T cells, but under the particular conditions he tested, if with certain tyrosine kinases expressed in T cells, PD-1 prevented tumor outgrowth in mouse T cells, post-thymic T cells. So our clinical trial is shown here. That was led by Ed Statmauer at Penn. The consort diagram showed 43 patients screened, and then 4 patients where we manufactured final products, and 3 infused. So the trial was really very difficult for a number of constraints that we had in the trial design because it was first in human and to really optimize safety. So we had many patients who screened out because they're A201 negative, and most because they didn't have antigen expression biopsy or other screening assays. And then some patients had disease progression before we could infuse them. We were required to culture the cells for a month after manufacturing to show that there was no growth factor independent growth of the T cells to exclude transformation. And that made the vein-to-vein time very long on this trial. So not an easy trial to recruit to. The first 3 patients infused are shown here. And 2 had myeloma and 1 had sarcoma, all very heavily pretreated. This patient 8 lines of therapy and 3 prior autologous stem cell transplants. This patient had 2, and a very heavily pretreated sarcoma patient. One good outcome of this was we found basically no cytokine release syndrome, no acute toxicity from the infusion of these cells. That was a potential risk because we found in our previous zinc finger nuclease trial that there were responses to patients after the engineering. And since Cas9 was bacterially derived and healthy donors all have responses, both humoral and at T cell levels to Cas9, we didn't know if this would trigger infections or adverse events immunologically mediated. We did not see that. And we then spent a lot of time analyzing the infused products in the patients, in this case with digital droplet PCR, so that the cell that we infused had up to 4 genetic modifications that could either occur independently or be assorted together, because the genes that we targeted, the alpha chain of the T cell receptor, the beta chain, and PD-1 are all in different chromosomes, creating the possibility to have rearrangements and translocations. And then PD-1 was then hopefully knocked out, and then we had the transgenic T cell receptor that would be expressed in the place of the endogenous T cells. We had in our negotiated with the FDA that we used a so-called Goldilocks approach. And in the first patients, the goal was not to achieve maximum editing, which we could do at about a 90% efficiency, but we used less guide RNA complexes and had a 45% editing of 4 different final products here for the alpha chain of locus, and then 15 to 20% PD-1 in the beta chain of the TCR. We found in preclinical studies that these edited transgenic TCRs function better as far as killing tumors that were expressed in New York ESO1 TCR. And these are the 4 final products with the average lysis by looking at killing tumor cells that are a melanoma line that's HLA-A2 positive and expresses New York ESO1. And if we, this is the killing, very potent killing was observed with the aliquots of the infused cells in the patient. These were cells where we mock electroporated and the cells retained their endogenous T cell receptor. So these killed at a lower effector to target ratio. We think because there's less of the mixed dimers that can be created by having the endogenous T cell receptor pairing with the transgenic T cell receptor. We had a sensitive assay that Simon Lacey made for Cas9 protein that could detect femtograms of Cas9 per cell. And that was easily there early on in the culture detectable. It became undetectable before the cells were harvested and infused. And we looked both for humoral immunogenicity to Cas9 and at the T cell level, and basically found there were pre-existing levels of immunity to Cas9 in the 3 patients who were infused, but up to 6 months later, we didn't see a bump in those titers. So it appeared the cells were non-immunogenic as far as Cas9, at least in these very heavily pretreated patients. I would caution that, we didn't test multiple infusions of cells, and it's possible that trace amounts of Cas9 could, over multiple exposures, sensitize patients and then have immune reactions, and potentially that could impact engraftment of these cells. We also looked for off-target mutations. And Rick Bushman adapted the assay called GuideSeq that Qi had made. This is an assay where oligo DNA is added to cells at the time of Cas9. And then with barcodes, it gets adapted during DNA repair. And then you could PCR and identify where breaks were in the cell and find, are they associated with guide targeted cuts or are they off target? And Rick improved the signal to noise ratio of this assay by increasing the length of the ODN. And that's the assay we used to look at the samples infused in the patient and then later in them. So of the 4 final products that were made, we found different off-target editings and on-target efficiencies between PD-1, the alpha, and the beta chain of the TCR. So there's an average of 99.4% on-target editing even with this very first generation Cas9 that we used. So very highly specific in patient 07. I mean, we couldn't find any off-target edits at PD-1. And the worst guide was the beta chain of the T cell receptor, where it was about 96% on target. In detail is shown here, say for patient 39, who was a sarcoma patient. This is the guide RNA here and the PAM sequence of, over almost 8,000 reads that were on target. There were just a handful of off target. This is, for instance, a chloride channel. And this is the guide where the off target edits were. There were 6 reads and a long non-coding RNA. Neither of these have any known function in T cells. So, you need long-term follow-up on these, but we found in general that these did not have preferential survival in the patients, rare off-target edits. And we also looked at a more macro level for chromosomal translocations and rearrangements. And with the 3 guide RNAs targeting 3 different chromosomes, there's 12 possible translocations. We found that those were detectable this is during manufacturing. So from day 5 after electroporation of the guide RNA and Cas9 out to just before harvesting, they went down on this log scale as far as frequency. So between 2% down to, point level of detection of this assay. So they also did not seem to have a gain of fitness because they decreased in time in frequency in vitro and also in the patients. So in the 3 patients who were infused, we found here out of a month and 6 months, decreasing 3 to 6 months later, decreasing efficiency on a log scale. So the translocations were not random. They varied depending on which potential translocation they were. And in all patients, they decreased in frequency over time, suggesting that they don't convert a fitness advantage. So we found that these were present in this is probably the most concerning aspect of this technology with multiplex genome engineering but when you look in the literature, there is data that healthy individuals, presume 50% of us on this call today, have rearrangements in our peripheral B and T cells. And they occur in sites known to be leukemogenic. So they're not necessarily a health risk. And therapeutic radiation and gamma radiation, either from for clinical treatments shown here or prolonged space flight, induces B and T cell translocations that you can easily see by FISH and other assays. And in the previous trial using TALENs done in London, where 2 genes were targeted, they found 4% of the infused cells had translocations. So we found about 1% of them in our trial, and this is an issue that other approaches such as base editing may get around, where we don't break DNA. So this is then looking at the engraftment of these cells in the patient. So our T cell receptor has a WPRE sequence in it, which we can assess for by PCR, and we found very stable engraftment in all 3 patients. It varies by an order of magnitude on this log scale of copies of the TCR per T cell over time, but out to 6 to 9 months, we found very stable engraftment. And also the T cell receptor cells traffic to the tumor. So in red is the 2 myeloma patients, and these are bone marrow biopsies and looking for the transgenic T cells. And this is the sarcoma patient. So in all cases, there were T cells expressing the TCR in the tumor microenvironment. We also found evidence in the 2 myeloma patients, evidence for T cell receptor editing, and I'll get to that. So of the tumor. But what we found here is that the survival of these 3 patients was much better than the previous trial that we reported in 2015, So these are myeloma patients given CAR TCR cells targeting NeoRKISO1, but without editing of the endogenous T cell receptor. And here the half-life of the cells is about a week in patients, with some patients having long-term survival at low frequencies. And here we found in our 3 patients, the average half-life by modeling was about 84 days in the 3 patients, and just very stable. And I mentioned that there was editing not only of the T cell receptor being edited out, but in the tumor microenvironment. So in patients 35 and 39, we found that the cells could be retrieved out of the patient, and if they functioned well, so this is stimulating the cells with cells that having the peptide, and they grow out progressively out of patients. So standing by the V beta 5 8.1, which is what this T cell receptor had, they preferentially expand, and then they kill at very low effector to target ratios. So they did not appear exhausted in these patients. They retained function for 6 to 9 months in the patients. And this just shows that the patients, the survival of the PD-1 transgene knockout was not enhanced in these patients. We had worried maybe because of that paper about transformation that could lead to that. And what we found out is exactly that the cells having PD-1 edits were actually selected against in vivo in these patients. There were no clinical response in these patients. All 3 had stable disease at a month. This patient died in December, and this patient had in the sarcoma mass about a 50% decrease that persisted for 4 months, and then he began progressing. And we could look at the tumor microenvironment in the bone marrow of the myeloma patients and look at the genes targeted by the T cell receptor, which were NeoRKISO1 and LAGE1. We found that disappeared in this patient after infusion. And the green line is the T cell receptor transgene. So the patient had trafficking to the bone marrow and edited out of the antigen. It was transient in this case, but this is classic data to support that there's T cell editing of the tumor antigens expressed in the microenvironment. Unfortunately, the patient continued to have myeloma. It just became NeoRKISO1 negative. So it's not a driver oncogene for those tumors. We looked with Howard Chang's lab at Stanford at single cell RNA of these cells, both in the harvested product and in cells recovered from the patient using 10x genomics, but modifying this pipeline to look also for the transgene sequences, as well as all the genes expressed and then mutations at the sites of guide RNAs. And if you look then at the infused product, we found that about 80% of the cells infused product had an alpha chain mutation around the guide RNA. And between 10 days and 104, basically 4 months later, it decreased, but it was stable between 10 days and 4 months for the alpha chain and for the beta chain of the TCR. We found about 15 to 20% of the cells had mutations around PD-1, and that basically declined over time in the patients out to 4 months. We can make a Venn diagram then and ask this very interesting question: are these mutations independently assorted or not? They're all in different chromosomes. So this is 27 different cells from in patient 39 that were in the infusion product. And there were some cells that had only mutations in T cell receptor alpha chain or PD-1 or beta chain. But 6 of the cells had mutations in all 3 targeted genes and also had the sequence for the NeoRKISO TCR. So these had, of the 16 possible mutations, had all the desired mutations that we wished. We found that survival of these triple edited cells, so at the single human genome level, about at baseline, 10% of the cells had all 3 mutations, but that decreased in frequency at 4 months in vivo in the patient, and whether or not they had the transgenic T cell receptor. And we found that, and just looking now at the PD-1 of the, we found in our preclinical studies that PD-1 knockout cells function better initially. And we found about 25% of the cells that had the transgene also had a PD-1 knockout, but that decreased at 10 days and 4 months later, so it was selected against. Turns out this is what John Weary and Nick Haining had shown previously in mice that had given adoptive transfer of knockout cells and then a chronic infection with LCMV. The PD-1 deficient cells initially function better, but then don't transition to memory. And I think that's consistent with this data we see here. And the cells, though, were able to show by these UMAP products, again in patient 39 at day 0 to 4 months, that they're healthy cells, T effector cells. So this is the gene, the New York ESO1 transgenic TCR, and that co-maps with TCF7. And TCF7 is probably the gene best associated with long-term healthy memory T cells. And the 10x genomics is shown here for patient 39. They evolved over time in the patient and did not show a pattern of exhaustion. So for instance, this is the IL-7 receptor, which was low in the infused cell, and TCF7, which I just showed you at the single UMAP plots. It's enhanced when we look, at the cells in the blood 4 months later. So this is good news that they appear to be healthy memory cells that have this TCR signature. So in summary, we found that these CRISPR-edited cells were safe as a single infusion. That in spite of the very heavily pretreated status of these patients, they had there were no side effects, and it's feasible in manufacturing the cells. I think this has, for the field, numerous potential follow-ons that could be done to with many different genomes, but basically now that could be targeted, enhance the function and specificity, and change the therapeutic index of infused T cell receptor and CAR T cells. And with that, I'll just skip to the end and acknowledge my lab and the people associated with this on the CRISPR trial and the Stanford group and the most recent data with the CRISPR engineering, the single cell sequencing. And with that, I think I could take questions, and I'll stop this.
Thank you so much, Carl. It was fantastic. I've been looking through the questions as they come up, so I'll just kind of read them out. It's just a selection of them. One is, why do you think there was more knockout of the TCR alpha chain compared to the TCR beta and the PD-1 genes? Is it because of guide efficiency, and was it done by design?
So that's a legacy going back to 2016 when the software and it was basically an inefficient guide. We now have better guides, and we can target both the alpha and beta chain at 90% efficiency, with improved T cell receptor beta guide. So that's why it had a higher incidence of off-target and less on-target efficacy.
Would you want to target as a follow-up, would you want to target the PD-1 more, given that, or more efficiently, given that it seemed to be selected against?
So, what I think is, and I think, what we want is a set of cells that have both very potent acute effector responses, and then can set of cells that are poised to become memory cells and do immunosurveillance. And so I think we want basically, whether that could be one cell or you have a cocktail cells that you infuse. I think what we found, and consistent with the mouse literature, is you don't want all of them to be permanently disrupted at PD-1. I mean, you could have switches, you could have, to toggle it on or off, and various strategies for that. But if they all can never express PD-1, I don't think that you'll get a good memory population of cells. So this is fortunately, we have the technologies to improve that.
Great. Another question somebody named Bill asked, is there a P53 response during the multiplexed CRISPR modification in the patient's T cells?
That's a great question. I think there is. We actually are exploring expressing dominant negative P53 to interrupt that for a short time after electroporation so that survival will be better. And Luigi Naldini's lab in Milan has done some of that in stem cell engineering. So there may be other chemical inhibitors, et cetera, that could do this as well. So that's the exciting part about this is there's many ways. I mean, our efficiency could be much, much better, using today's tool set in the lab. The barrier is it takes quite a bit of time, to get it into clinical trials.
Right, right. One other question about the loss of antigen expression. You mentioned that the myeloma tumor came back with loss of NYE. So did you see any tumor antigen loss in the sarcoma patients?
Well, that's a really good question. So, molecularly, we know sarcomas express via different mechanisms and translocation than sort of a lot of epigenetic reprogram, which is primarily what I think happens in myeloma. And so in other studies, not our own, people have found that, for instance, done by in Seattle and where they've used with Adaptimmune, they've treated with New York ESO1 T cells that did not have edited TCRs, but they found that in the sarcoma patients, they continued to express the New York ESO1 gene, unlike what we found with myeloma. And Crystal Mackel has some similar data at Stanford where it appears that in sarcoma, they have another mechanism of escape, whether it's exhaustion of the T cells or downregulation of class 1 process machinery, I think is unknown, but at least by expression of the gene New York ESO1, the sarcoma patients tend to do that. We were not allowed to biopsy our patient. We had one biopsy of a needle biopsy of that mass, but we don't have enough cells to know molecularly if New York ESO1 was still there.
And just since we're out of time, maybe I'll just ask the last question to kind of tie things in here. Do you have any thoughts about the cytokine release syndrome in COVID?
Yes, so we I actually have at the end of the month a perspective that will be in Science on this. There are international trials testing this. When you look at the cause of death in COVID, we don't know much, because pathologists have not been doing many autopsies because of BSL-3 and really afraid of biohazard exposure. But it looks like many patients with severe COVID get HLH, hemophagocytic lymphohistiocytosis, or macrophage activation syndrome. So it looks like it's macrophages and monocytes in not the same as what you would see in the ARDS with bacterial sepsis in patients with very severe COVID. And there are anecdotal reports that blocking the cytokine release makes us better in Wuhan. So controlled trials are being done now with a variety of reagents from anti-IL-6 receptor to anti-IL-6. Steroids so far are reported in the case of other beta coronaviruses like MERS and SARS, actually to make the disease worse. It's controversial in COVID whether or not steroids are useful at some point in the disease or not. But so I think clearly there's a lot of work to be done here to prevent the immunopathology of COVID, as well as obviously to get vaccines and active antiviral agents.
Right. Great. So it's a little after 1 o'clock. I just want to mention to all the participants that who've asked questions, Dr. June will have access to these questions, and he may follow up during his convalescent period. Also, just as an FYI, there's no talk tomorrow, but Dr. Slack will be speaking on Thursday. Thank you so much, Carl, and good to see you, and take care.
Thanks, Marcella, and thanks, everyone. Stay safe.